last part of this topic, that is spermatogenesis and oogenesis. So in that we have to be seen what is meant by spermatogenesis, oogenesis, what is the different process, what are the difference between that both of them, that is spermatogenesis and oogenesis. First upon we have to see this diagrammatic representation of spermatogenesis. So in that, spermatogenesis means that is the process by which the male gametes known as a sperm is formed. In this process, each sperm is haploid, which is containing single copy of each chromosome. Okay. In order to create the haploid gamete, a cell undergoes the process of meiosis, in which genomes is replicated and divided twice to produce the 4 haploid gametes. This process generally occurs in semiferous tubules of the testes, which of the following different stages. So it is followed by the maturation in the epididymis, where they are secreted in the form of semen along with granular secretion. As yet clear? Okay. This process begins during the puberty and ends only when the individual dies. Complete process of spermatogenesis in the male are carried out by the action of Leydig cell, that hypothalamus and pituitary glands. The quantity of these sperm gradually reduces with the age and finally leads to infertility. Okay, so see this diagrammatic representation. This is the diagrammatic representation, that is how sperms is produced. Okay, so see these sperms, then another germinal cell. So in that diagram, given number on that specific organ at the time of spermatogenesis. So see the first number organ, that is a spermatide. Okay, spermatide. Then second, see, okay, above one, this one. This second is a germinal cell. Then below that, see that third. Have you seen first, second, then third is the epididymis, then fourth, okay, is that clear? The fourth one is vasa deferens, then fifth number is testis, then sixth number coiled seminiferous tubule. These are the, how much you have seen in your diagram, that is 4. Okay, then seven number, that tube like, that is a cross section of seminiferous tubules. So what is the purpose of that spermatogenesis? The process of spermatogenesis occurs to create mature male gamete, which then these gametes which then fertilize female gamete to create a zygote, a single celled organism. This results in a cell division and multiplication to create a fetus. For a healthy offspring, a number of chromosomes must be maintained properly across the body, as a failure can lead to some abnormalities. Okay, so this one is also this 23, that is 4 sperm. Okay, that is a haploid. In human being, how much pairs of chromosome? That is 23. That means how much chromosome? That is a 46. That was sister chromatid at the time of meiosis. This spermatogenesis, 46 single chromosomes, it can be replicate in interphase. These are divided into 2 phases, that means cell division, meiosis first and meiosis second. This 46 sister chromatid is divided into 2 daughter, that means 23 sister chromatid, 23 sister chromatid. Again, these 23 sister chromatid is divided or cell divide into in the phases of meiosis second, and it could be given 4 daughter cell, that means 23 single sperm. Is that clear? Okay. So in first stage, second stage, third stage, what is going on or what is happened? In first stage, the diploid spermatogenia is situated in the seminiferous tubules, which include twice the total number of chromosomes. This replicate mitotically in the process of interphase before the methods of meiosis first and create how much pair? That means 46 pair of sister chromatid. At the time of second phase, this phase, the chromatid allowed to exchange of genetic information through the synapsis process, that means synthesis process. It is done before dividing into haploid spermatocytes through the meiosis. Then in case of third stage, this stage of cell division, the two new daughter cell will be further divided into 4 spermatid which is having unique chromosome that are approximately half in a number to the original spermatogony. Okay, and fourth stage. So at that fourth stage of cell division, the cells move from the lumen to the testes to the epididymis. They get matured there and develop into the four sperm cell with the growth of microtubules on the centrioles to develop an enzyme. Okay, so there are different factors that can be affecting on spermatogenesis. So this spermatogenesis process, it is a very sensitive and that can be easily affected by following different factors. So which are the factors they have affecting this very sensitive process? In that minute change in our hormonal level. For example, testosterone is developed through the hypothalamus, leading cells and pituitary gland. Then this process is very sensitive to change in a temperature. Then another factor that could be deficiency in your diet. Then alcoholism. Then exposure to different types of drugs. And last, presence of a disease can affect the rate of sperm formation adversely. So these are the different factors which can be affecting the number of or process of spermatogenesis. Then last one, oogenesis. That means oogenesis is also called as a ovigenesis. So what is meant by oogenesis? It is the formation and maturation of a female gamete. In case of spermatogenesis, that is a male gamete. Is that clear? And oogenesis, that means formation and maturation of a female gamete. In oogenesis begins or start in the prenatal period. In the female fetus, the germinal epithelium forms into the cluster in which one genocytes, which can be differentiated into an oogenum, which are containing typical cell constituent or component. The oogenia then undergo prophalia or proliferation, which can be prior to shortly after the birth, resulting in the flinked ovaries, which are containing the sole reservoir of all future ova, which can called oxide, oocytes. Okay, the growth of oocytes is characterized by the enlargement of cytoplasm by accumulation of a different size of granules, dinoplasm or yolk, that is called as a yolk. The development of an egg's membrane, which is called as a zona pelluda. Then mitotic proliferation of follicular epithelium adjacent tissue. There are two stages in the growth of oocytes, the first stage and second stage. In first stage, the growth is rapid and intimately associated with the development of ovarian follicle. Then this attainment to its mature size that could be occurs at about the time of anstrom formation begins to the follicle. And what is happened or what is going on in the stage second of octet, that means in octet does not grow in size while the ovarian follicle responding to pituitary hormones, which can be increased very rapidly in their diameter during the latter phase of follicular growth of oocytes, which can undergo maturation. And this is also diagrammatic representation of this oogenesis. Okay, we have seen that primary octet, that means this consists of a 46 single chromosome which can be replicated in the process of interphase in mitosis, in case of meiosis first and meiosis second. So they can be divided into two daughter cell or sister chromatid, that means 23 sister chromatid. Okay, then these meiosis first and meiosis second get divided and it can be given single ovum in, that means 23 single ovum. So the this consists of a three polar body. And last one, most important part, that is sometimes asking in your examination, what is the difference between spermatogenesis and oogenesis? In that some differences are explained here. So in case of spermatogenesis, where they are located, they can be their location that could be occurs entirely in the testis, and in case of it occurs mostly in a ovary. Then at the time of mitotic cell division, in case of spermatogenesis, equal division of cell it could be occurs. In case of oogenesis, unequal division of cytoplasm it should be occurs in case of oogenesis. Then another difference in both, that means germs line epithelium. So in case of spermatogenesis, it involve in gamete formation or gamete production, and oogenesis process it should not be involved in gamete production. So what about gametes? In case of spermatogenesis, they have to be produced 4 number of gametes, and in case of oogenesis, it should be produced 1 plus 2 or 3 polar bodies. Then size of gamete. In case of spermatogenesis, size of gametes, that means sperms are smaller than the spermatocytes, and in case of oogenesis, ova is larger than the oocytes. Then what is the timing of spermatogenesis and oogenesis? That duration during the spermatogenesis, that is uninterrupted process, and oogenesis in arrested stages. Then onset it should be start at the time of puberty in case of spermatogenesis, and in case of oogenesis it should be start in fetus or prenatal stage. Then release of spermatogenesis spermato, it should be, this process is continuous, and in case of oogenesis it should be monthly from the puberty in the form of menstrual cycle. And lastly, end of that spermatogenesis or sperm, that is lifelong. Okay, but the quantity or number it should be reduced with the age, and in case of oogenesis it should be terminate with the menopause.